portion of Nephrology Grand Rounds, which is Biopsy Conference. And I believe the first case will be presented by David.
That is correct. Thank you. Okay, good morning. Thank you for being here this morning. I would like to introduce Dr. Ambrose, who will give us the clinical information on this patient that this case we're presenting. And I'm not sure if Dr. Ambrose is ready yet.
Yeah, Dr. Ambrose joined.
Thanks.
David, the presentation is not in presentation mode. I don't know if you want to do that or.
Sure. Let me. I am usually there is an option to swap screens, but in this case, I'm just going to share my screen by itself instead of the window and see how it works.
That looks good.
Okay, great.
So I see Osama's joined with his iPhone.
Dr. Ambrose? Okay, I'm not sure if he's muted or not.
Hello? Can you hear me?
Yes, we can hear you, Dr. Ambrose. Yes.
Can you hear me?
Yes.
Oh, sorry. Yeah, this is a 63-year-old gentleman presented to my clinic for evaluation of CD5. And he has hypertension, previously uncontrolled, no regular follow-up, creatinine of 5, non-invasive workup showed, proteinuria of like 300 milligrams, and relatively bland urine sediment. And he had some anemia with hemoglobin of 8. Immunofixation showed monoclonal glomerulopathy. So we thought this is likely hypertensive nephrosclerosis. However, we want to make sure this is not myeloma. So we urgently referred to hematologist for evaluation.
Thank you, Dr. Ambrose. So after that information, obviously we received our kidney biopsy, and the biopsy we received was predominantly medulla with a scanned portion of cortex. You can see a few glomeruli here. In total, we received around 1 to 3 per level. But even with these, because of what I am about to explain in a few minutes, we were able to still make a diagnosis.
I apologize, David. Maybe before going into the pathology, now that you've shown the low magnification, maybe we want to give a chance to the audience to ask questions from Dr. Ambrose about the clinical history and also differentials. If you can share those with us in the chat box or just by voice, that would be great. Any questions for Dr. Ambrose about the clinical history and differentials?
Any cardiomyopathy or peripheral neuropathy?
So that's a good question. We went back asking details about any history, such as cardiomyopathy or neuropathy or any systematic manifestation of any kind of disease. it was not. And basically, other than central obesity and hypertension, patient has relatively good health. One piece of information also I need to emphasize that we asked extensively about family history of kidney disease, early death, and there was none. Basically, father and mother died of old age and no family history of kidney disease.
Dr. Ambrose, I have a question for the hematology workup. What was the results back by the time of kidney biopsy?
Yeah, actually, the kidney biopsy was prompted by the hematology evaluation.
Okay.
So there was no evidence of multiple myeloma, and still we have this immunoglobulin not fully understood, and then we proceeded with kidney biopsy.
I see. Thank you.
It looks like Brendan has his hand raised for a question.
What was the magnitude of the M spike?
I don't recall, but it was not very high, and the ratio was normal. And actually, when we first referred to hematology, we were still thinking that the hypertensive nephrosclerosis is the main differential diagnosis because of the history, prolonged untreated or loss of follow-up of high blood pressure.
Okay, differential diagnosis. Anybody wants to share with us what you guys think?
It could still be AL amyloid, but just with vascular involvement and ischemic glomeruli. Doesn't have a lot of proteinuria.
AL amyloid, I see that Bilal has mentioned electroamyloidosis. Okay, that's interesting. Anything else?
Is the patient Native American or Hispanic?
Yeah, as mentioned in the history, he's Hispanic.
I'm going to go with the LEC2 also because you probably wouldn't have mentioned family history so much.
Okay, let's look at the biopsy then. David.
Thank you. Thanks for those clinical insights. I apologize for jumping quickly on this. As I mentioned in our lower power view, we can see that this biopsy is predominantly medulla with a very scant portion of cortex. And in average, we had 1 to 3 glomeruli total. And you can observe, and I will show more high power views of these extensive interstitial fibrosis and tubular atrophy and separation of tubules from each other. So overall, this kidney looks with extensive chronic injury. Now, what's interesting about this is the expansion of the interstitium with this material that we can identify in these areas. It does not have a particular, let's say, texture to us as pathologists. Doesn't have a fibrillary character of it. It looks more glassy. And the tubules, as you can identify here, these tubules are dilated. Some other tubules are almost completely obliterated. This present a thickening of the basement membranes with corrugation. That it's an indicator to us that this tubule is undergoing atrophy. And these glomerulus, probably not the most photogenic one, but the capillaries are patent. So our attention should be, in this case, centered in the interstitium where we identify that the lesion involved in this kidney is. Now the interstitium is infiltrated by this pale amorphous material. It does not have cells being part of it. I mean, there are scattered interstitial cells that are present here, but they are not within the deposits. They're in the periphery of the deposits. So that's why we describe this as a cellular material. And we call it amorphous, as we said. No, we don't see any particular character or texture to it. Now, when we scan this, even though we have very few glomeruli in this biopsy, but the few patent glomeruli that we identified did not have involvement by this pale amorphous material. It was confined to interstitium, and some vascular structures were involved as well, and focally also involving some tubular basement membranes. And here's more detail of those, a little bit of higher power view. Now, easily, if someone can tell me, it's a pretty easy question. What can we use to further characterize these deposits?
Okay, so in the chat box, I read Congo red.
Perfect. That would be our go-to in this type of cases that we suspect that is amyloid. And indeed, this case, the interstitial deposits of cellular material and the corresponding area in a different level now stained with Congo red, it's positive in these deposits. Now, this helped us as well to see that these deposits are located in the tubular basement membranes and some vascular walls. When we put this Congo red stain under polarized light, we have this abnormal illumination that we call apple green birefringence, and some fibers are staining, some areas staining with this yellow color, and some areas staining by this bluish green color. This is not the real color seen on the microscope, but the camera probably won't capture what your eye can see. But anyways, what the important thing is the abnormal pattern of illumination in this case. Bioluminescence microscopy, very important evaluation, since we have in the differential that this patient may have AA amyloidosis. We had a total of 5 glomeruli with 4 globally sclerosed. This is further supporting the chronic damage on this kidney. The glomeruli did not show more than trace mesangial staining for IgM, which can be non-specific, and the tubular cast that stain for IgA, IgM, this is pretty common to be IgA positive, but most importantly, there's no significant staining bias for either kappa or lambda. So in this part of evaluation, since we had the history of an IgG kappa in this patient, we don't see that bias identified by the evaluation of the light chains. And in this specific case, we did not see significant interstitial or vascular staining or glomerular staining. By electron microscopy, these are the thick sections stained with toluidine blue, and we can confirm the findings. You see that this is a tubular basement membrane that is thickened and diffusely infiltrated by the same material that we see in the interstitium. The glomerulus identified in this area is widely patent. The mesangial regions are not expanded, and it's usually where we can more easily identify it in case of amyloidosis if there's something deposited there. In this case, we don't see it. And just the same thick section with the same amorphous material. This is another tubular basement membrane diffusely involved in here, and then the interstitium. Now, in an evaluation of the glomerulus, we see these glomerulus low power view in bioelectron microscopy. This is the mesangium, which doesn't show any increase in cellularity and no significant increase in the matrix deposition. Now you see the capillary loops that are widely patent. This is an endothelial cell. The podocyte food processes are preserved. We can see this as well in this other capillary loop. Likewise, there's some focal blunting and effacement of podocyte food processes, but this was not widespread. And higher power view of the glomerulus, we see that the endothelium is mostly preserved. There were some other capillary loops with a mild expansion of the subendothelial space. This is not depicted here, and it was mostly consistent with the history of this patient of having a history of hypertension, which can induce certain endothelial injury. And usually we can see that by electron microscopy. Now, importantly, what we see here is this tubular basement membrane. It's thickened. We see the epithelium showing loss of the microvilli of the luminal villi here. And there's a deposit that we identified surrounding the tubular basement membrane, pretty similar to the deposits that are present in the interstitium. We have a higher power view of this same area, and we identified that this has a fibrillary character to it. And when we see this at an even higher magnification, we identify these fibrils are randomly arranged, and they're not branching. They don't have other particular substructure. And the diameter is consistent, around 10 nanometers in average. So based on all of these findings, putting all this together, what will be your pathologic diagnosis? Only based on the findings that we can see at this time. I will open the chat box and see if somebody wants to.
Yeah, there's no
message over
response.
there.
Maybe it's obvious. Nobody is guessing.
Or what is not? What is the likelihood of us confirming amyloid of certain type based on the information, knowing that our immunofluorescence was negative? And at this point, we cannot fully rule out that this was not an AL amyloid, but the likelihood is lower since our immunofluorescence is negative. And so what we presented as a preliminary diagnosis was that this is a case of amyloidosis of undetermined type that was focally involving the tubular basement membranes, the interstitium and vascular walls. And what was important for this, and this is just a summary of what I explained, so I won't read this text, but importantly, there's no glomerular deposition of amyloid by light or electron microscopy or Congo red stain. And based with the clinical history of an IgG lambda monoclonal glomerulopathy was raising concern for the amyloidosis of primary type or AL type, we did not see any obvious monoclonal immunoglobulin or light chain restricted deposition in the renal parenchyma by immunofluorescence. And based on this, we decided with the oncologist to send this out for further analyzing these amyloid deposits. So our results come back, and the results from mass spectrometry, and this is reported as an ALK2 amyloidosis. So provided an addendum based on this and definitive diagnosis of an amyloidosis ALK2 type involving tubular basement membranes, interstitium, and vascular walls. So far, is there any questions regarding the pathologic findings before I move on to a very brief discussion?
Just a quick question. Do you see predominantly interstitial deposits of amyloid in other forms of amyloid, or is interstitial amyloid most commonly ALK2?
As far as I know, this is most characteristic of ALK2. I would say we can also find glomerular involvement, for example, in ALK2, but it's very rare. So if we can put a buzzword or something associated to the finding of interstitial of amyloid of predominantly interstitial location, this will be related with ALEC2. I am not sure if some of the pathologists, the renal pathologists would like to add something to this. I am not aware that other type of amyloid have a predominantly interstitial deposition in this case. Okay.
I have a clinical question, and it just really came out with, I have another patient, from Hispanic origin with elect amyloidosis, end up with end-stage renal disease at age 59. He has no history with uncontrolled hypertension. So it's just kind of like there is a variation in the severity of amyloidosis. I wonder if this patient, the one you are presenting, had controlled blood pressure with that degree of amyloidosis cause kidney failure at this age. That's sometimes that's at this point is not clinically relevant, but is also important when we are considering transplant, because how soon will that come back to the kidney? So that's a question for everybody.
Usually with ALEC2, the relapse should be, I mean, it develops very, very slowly. So I think this person, if he goes through a kidney transplant, should have a good long-term outcome. So unlike the AL types, this should have a better long-term prognosis.
Thank you. Thank you. I will confirm Dr. Malik's information. Of course, it's considered somehow slowly developing or relatively benign type of amyloidosis. And I will explain in another slide one of a few risk factors for rapid development. Now, this was described in 2008. I mean, not the molecule itself, but related to amyloid. On this was described in this paper. And this is located, the human LEC2 gene located in chromosome 5. The molecule has different functions. Initially, it was thought to be involved mostly in repair process, chondrocyte development, but later was discovered that this was a modulator also of immune inflammatory reaction and metabolic functions, mainly synthesized in the liver. This is particularly common, this type of amyloidosis in Hispanic patients in the Southwest United States, but also has been reported in a higher proportion in people of Egyptian descent, mostly of papers that are coming from Europe, where a higher proportion of an immigrant population in these areas are present. Now, the renal tissue is the major target, and usually a low-level proteinuria and varying degrees of reduced GFR are present. There's diffuse cortical interstitial amyloid deposits, and interestingly also, there's liver involvement, and frequently an incidental finding. So regarding this, I am not sure how deep should you look for liver disease in this patient, but it definitely happens at the same time as renal involvement. So it will be worth it to see how the liver is functioning. The etiology is not, there's no definitive answer to it. Some information of an undefined genetic mutation, especially GG genotype, or also hepatocellular damage. There is some data that is suggesting that patients with diabetes have higher titers or higher levels of LEC2 amyloid in serum. And overall, this could be a marker of hepatocellular damage. This is upregulated by the hepatocytes, and the interaction of this molecule with the local molecules in the matrix or increased local concentrations may lead to the stabilization and formation of fibrils in the renal interstitium and create this clinical information. I'm running out of time, but most importantly here in this slide, just to remind us that just based on this, it's important to check our patient's diagnosis if there's any doubt that this may not be an AL amyloid by laser dissection and tandem mass spectrometry studies. Now, this is what I was trying to explain. This is a case series in a European center, 24 patients, mostly of Egyptian and Southeast Asian descent. The median estimated GFR at diagnosis was 33 ml per minute, and the proteinuria also low, non-nephrotic range is 0.5. They were followed up for about 4.8 years. From those 24 patients, 4 patients die and 4 progressed to end-stage renal disease. Now, in this other study that I present here, what they found most important, this paper was based on 72 patients, and the median renal survival was 62 months. And 1/3 of these 72 patients developed end-stage renal disease. And the best predictors for development of end-stage renal disease was the percentage of global glomerulosclerosis, the presence of diabetes, and the creatinine level with a threshold or a best cutoff of 2.0. And this graph representing the patients that had ALK2 with no other concomitant morbidity, no diabetes, no hypertension, and presented different levels of creatinine. The ones with creatinine more than 2 obviously had a higher rate of progression towards end-stage renal disease compared to the patients who did not have that level of creatinine. So this is what I have. I only have 2 minutes for questions, but I'll be happy to take any questions right now. For me or for Dr. Ambrose? Okay.
Thank you very much.
Thank you.
Dr. Ambrose and David.
Thank you.
Any questions, comments? If there is none, we move to the second part, which would be presented by Dr. Akhilesh, and that will be the case that he discussed at ASN Biopsy conference. So especially for those who did not have a chance to attend that, that's a very interesting and educational case. Thank you.
Thank you, Behzad. So this was a case that I presented at the renal biopsy conference at ASN. It's actually a case that Dr. Smith, Kelly Smith, presented earlier on last year, but there's a reason we're revisiting this case because we've learned a lot more about it, and we've got some new tools that I wanted to show to everyone here and just kind of demonstrate the power of some new technologies. So let's get started. So I'll be presenting kind of both the clinical and pathologic findings in this case. But both Dr. Shannon and Koirala have interacted with this patient and followed this patient previously, though I think he's now followed in an outpatient setting. So this patient at the time of presentation last year was a 45-year-old male, originally from Mexico, but had a pretty complicated medical history. So he had hypertension and also had atrial fibrillation, which was complicated by a remote right basal ganglia hemorrhage with some residual deficit as well. So this is 10 plus years prior to the current presentation. And so, but the blood pressure was now pretty well controlled with the ACE inhibitors and kind of residing in the 120s to 80s or so for blood pressure. He had also experienced a deep vein thrombosis 17 years prior. So he had a IVC filter placed and was also anticoagulated with warfarin. So you can see now this is kind of 2 different things going on with both, hemorrhagic injury in the brainstem, as well as also with the anticoagulation requirement. Now, in terms of kidney disease, 3 years prior to the presentation, the current presentation, he presented with subnephrotic range proteinuria, about 2 grams or so, without evidence of active sediment. And this is in the context of obesity. So his BMI was about 40 or so, and his creatinine at that time was about 1.7. So it had been slowly creeping up. And so a biopsy was performed at that point about now 4 years ago for subnephrotic range proteinuria. Now, this biopsy showed some, again, cortex and medulla. There was already evidence of some scarring and injury. You can see here by this black material of this low power view. You can see that there are some areas of atrophy as well, some kind of microcystic dilatation and atrophy of some of the tubules. But we can take a closer look at this glomerulus over here. And we can see that it has a very characteristic appearance. So anyone in the chat or want to shout out what we think is going on with this glomerulus? Should be fairly obvious. Okay. Or maybe it's not obvious. So what we're seeing here is a glomerulus with some patent capillary loops over here. But in this segment here, the capillary loops look a little bit compressed or collapsed. So there's a buzzword there for you. And you can see that these epithelial cells look very reactive, and some of them contain these bright protein reabsorption droplets. So this is a form of FSGS, and particularly we're going to call this a collapsing glomerulopathy. And so, so about 4 years prior to the current presentation, this patient had a collapsing FSGS. So that was the diagnosis that was originally rendered. He was further evaluated for additional, viral potential causes, such as, HIV, and everything was negative. And so there was also some discussion about, how confident we were in this diagnosis, since there was a very tiny sample. But in either case, this at least provided an explanation for his proteinuria. Though typically with collapsing FSGS, we think about it in terms of a more aggressive, higher degree of proteinuria than the non-nephrotic range proteinuria that this patient was experiencing. So since that diagnosis, his creatinine has slowly been creeping up. So he's estimated loss of about 5 mils per minute per year. So his creatinine baseline had risen to about 2 to 2.2 or so. And this now all kind of got derailed last year in spring when he contracted COVID-19 infection. His COVID symptoms themselves were actually pretty mild. He had only mild upper respiratory symptoms, but he actually presented because and was hospitalized because of acute kidney injury. His creatinine actually peaked at over 12. He required dialysis. And it only went down to about 6 milligrams per deciliter at the time of discharge, at which time he was also COVID PCR negative. He also, at this time, had nephrotic range proteinuria. It was 4 plus. And this really didn't improve even after he was discharged from the hospital. And so about 6 weeks after this initial, acute presentation with COVID and acute kidney injury and, worsening of his baseline kidney function that was persistent now requiring dialysis, a biopsy was performed really kind of to see, okay, what's the current state of affairs in the kidney and what kind of prognosis was he going to have, okay? So you can see some of the lab values at that time. His creatinine was really, 12, 13 or so, with a lot of protein being spilled into the urine. So this is the biopsy we received, and this is the case I presented at the biopsy conference at ASN. So this was now in early 2020 when we received this biopsy. So again, we had what looked like initially a generous core of renal cortex and medulla. But there were only a very few glomeruli, only 3 to 4, and about 1 of which was completely sclerosed. And interestingly, there was only moderate tubular atrophy and interstitial fibrosis, again with kind of microcystic changes. And I'll show you some of these in a little higher power. So kind of interesting that the kidney function was, as severely affected, even though there's still some residual parenchyma, at least on the biopsy. So if we take a closer look, we can again see these areas of atrophy and scarring. The tubules are not big and plump and sitting back to back. Instead, we've got atrophic areas. We've got some evolving interstitial fibrosis. We've got some of these larger dilated tubules, which are undergoing what we call microcystic atrophy over here, filled with pink cast material. We'll take a closer look at some of these tubules, which are also acutely injured. And again, we've got a few glomeruli to examine. So at a higher power, we can see some of the key findings in the tubular interstitium. First, we'll start with a very normal looking glomerulus down here. So that's patent, no abnormalities there. We can see the acute tubular injury, as David showed in the previous case as well. We can see an attenuation of the epithelial cell layer with apparent widening of the lumen. We can see some areas of atrophy here. The tubules are pretty much almost disappearing over here and replaced by fibrosis. Okay? But not all the glomeruli were normal. Again, we had another glomerulus kind of with some collapsing type features if we were to stretch it out a little bit. So we have a segment that is a little bit collapsed. We've got some prominence of epithelial cells and even some foam cells in some of these capillary loops. So perhaps not as photogenic as the prior diagnosis of collapsing glomerulopathy, but it nonetheless seemed to have some collapsing type changes in the current biopsy as well. The immunofluorescence was negative. The EM, however, showed some additional findings. So clearly, when we think about FSGS, minimal change disease, the nephrotic syndromes, we think about diffuse podocyte foot process effacement, and that was certainly the case over here as well. So again, just to orient everyone, we're looking at a couple of capillary loops over here. Here is the basement membrane of one of the capillary loops, architecturally unremarkable. Here's another one over here. Here's a little central portion of the mesangium that's holding everything together. Nucleus of an endothelial cell over here. You can see that the podocytes on the outer aspects of these capillary loops have very effaced foot processes. They're very flattened. We don't see distinct separated foot processes whatsoever. We can also see perhaps the beginnings of some microbullous transformation and even pseudocyst formation within the podocytes, all markers of diffuse injury to the podocytes, correlating with the proteinuria that the patient was experiencing. In addition, though, in some capillary loops, we saw expansion of the subendothelial space. So this was a little different than the prior biopsy, which just showed some effacement of the podocyte foot processes. So in this capillary loop, we can see the endothelial cells actually look a little swollen. They're not nice and flat with their intact fenestrae. Instead, the cytoplasm is a little bit more plumper and swollen. In addition, they should be sitting directly on the basement membrane over here, but instead they've been lifted up because there's this accumulation of this more electron-lucent material. So this is what we call subendothelial space widening, and we associate it with acute endothelial injury or thrombotic microangiopathy. So interestingly, we saw this only ultrastructurally. We did not see any evidence of thrombotic microangiopathy by light microscopy whatsoever. So in this context, he got the diagnosis of collapsing glomerulopathy. This is in the context of recent COVID-19 infection. This is 6 weeks prior is when he experienced COVID symptoms. He also had acute tubular injury. There was some patchy inflammation in the interstitium as well that I didn't really show you here. There was also ultrastructural evidence for some endothelial cell injury. And of course, there was the chronic injury as well. So now the patient remained dialysis dependent after this. And there was a question. So remember, he was originally from Mexico, but is presenting with a collapsing type glomerulopathy. And so Dr. Shannon performed APOL1 genotyping in this patient. And he was found to have the characteristic 2 amino acid deletion that characterizes the G2 high-risk variant of APOL1. And this patient was a homozygous G2 for APOL1. So had a high-risk APOL1 genotype, which of course we know is associated with collapsing glomerulopathy. Okay. All right, so at this point, I presented 2 biopsies. The first biopsy for this patient was read as collapsing FSGS. The second biopsy was also read as collapsing FSGS. And so I'm going to preempt the thought in your mind, really, have we really learned anything new, right? I mean, so why am I even presenting this case? So what? So he's got a high-risk APOL1 genotype. He's got collapsing FSGS. We know now that COVID is a risk factor for it. But let's dive into this a little bit deeper and see what we can learn. So clearly, when we think about collapsing FSGS, and this is a nice review by Martin Pollack, collapsing FSGS, of course, has got a number of potential risk factors that can trigger it. So clearly, the original identification of APOL1 contribution to a disease, some of the earliest cases was, of course, related to HIV infection. We'll talk a little bit more about APOL1 in a couple of minutes here. And of course, in the last year, we've figured out that SARS-CoV-2 is also a potent interactor with high-risk APOL1 genotypes to trigger this collapsing type phenotype. We and others were some of the first to show that this was the case. And then when we think about COVID disease, now we're pretty much taking it as dogma now that COVAN, or COVID-associated nephropathy, is characterized with this constellation, not necessarily in all patients, but can be characterized by acute tubular injury, collapsing glomerulopathy, and also endothelial injury or thrombotic microangiopathy as well. And this patient really had all three of these features: had acute tubular injury, had a collapsing type FSGS in the setting of APOL1 high-risk genotype, and also, at least electron microscopy-wise, had some evidence of acute endothelial cell injury as well. Now, when we think about APOL1, of course, this was identified a little over 10 years ago by Martin Pollack and Jeff Kopp and others. And so this is clearly a very high risk for a variety of kidney diseases. And the hazard ratio, or the odds ratio for viral-induced disease like HIV is extremely high. And so we know that APOL1 high-risk genotypes are, of course, concentrated in West Africa, and also track along to the Caribbean populations and the American Black population as well. And so I told you that this patient was actually originally from Mexico. And though the prevalence of the high-risk homozygous genotype in Mexico is very low, it's still about 0.1%. Versus about 10 to 15% in the American Black population. And so this patient really already had a high-risk genotype, perhaps contributing to the original diagnosis of collapsing glomerulopathy, but was also perfectly poised to interact with now a more recent COVID infection to re-trigger, collapsing type presentation in the current case. Now, there's been a lot of debate about whether SARS-CoV-2 actually infects the kidney. Using proper reagents, in our experience and many others, I think it's becoming clear now that it's not even if it occurs, it's very, very unlikely. We've seen that even in autopsy lung tissues, that it can be really hard to find the actual virus, whether we use RNA detection methods or immunohistochemistry. And even in cell culture models where we have high rates of infection, it can be difficult to detect the virus because not all cells may be actually infected by the virus as well. And the same goes through with electron microscopy as well, that you've got to be very careful in how you interpret whether something is a virus or not by EM. So let's return now to APOL1. And so there's been a lot of study in this in terms of its contribution to HIV-associated nephropathy in Black patients. Much work has been done to show that APOL1 can be induced by immune activation, and particularly in terms of the context of interferon. High levels of circulating interferon are known to cause upregulation of APOL1. And there's been this really nice, complex mouse model developed by David Friedman and Martin Pollack, where they actually show that in the endogenous human gene context, there's marked upregulation of APOL1 by interferon, and this can cause kidney toxicity and almost collapsing type FSGS phenotype. So really what, at this point, we thought, okay, we understand the system, right? So APOL1 can be toxic to the podocytes. APOL1 is induced by interferons that can be seen in a variety of states. For example, lupus, systemic lupus erythematosus, or viral infections such as HIV, or now more recently COVID, right? So this is really what's going on in this patient. He has a high-risk APOL1 genotype. He got infected with SARS-CoV-2 that triggered a high interferon response and upregulated APOL1 to re-trigger his collapsing glomerulopathy, right? But is that really the case? Let's so hold that thought in mind, and we'll come back to it. So really what I'm trying to drive at here are what are the mechanisms that drive collapsing glomerulopathy and what's been done in this field. And so I see a comment in the chat from Ian DeBoer that malaria and APOL1 high risk also seems to be a good trigger for collapsing FSGS. Yes. So many infections, not just viral infections, can be associated with this. And again, we can think about it, you know, coming to a common mechanism of perhaps interferon-induced upregulation of APOL1 in the target cells. So really, this is a collapsing glomerulopathy is a difficult entity to study using molecular techniques, because as I showed you in this biopsy, very few glomeruli may actually have that particular histology. And even 1 or 2 glomeruli with that histology is sufficient to render a diagnosis of collapsing glomerulopathy and carries with it the whole weight of that prognosis in terms of, you know, a more aggressive disease course. Nonetheless, this really nice study about 11 years ago done by Jeff Hodgin and the Dagadi and colleagues used laser capture microdissection to try to address this issue. So for those who are not familiar, laser capture allows you to basically extract, physically remove a portion of the tissue that's affixed to a special slide, and then profile the gene expression, for example, in that particular captured tissue. So you can see in this study, they were interested in glomeruli, so they were able to selectively draw a circle around it and extract it for gene expression studies. But the problem with this is that it requires a lot of input material. And so they had to pool about 20 to 25 cross sections from each case to actually get enough material to evaluate. And as a result of this, they had to pool everything. So both normal looking glomeruli, the collapsing glomeruli, all of them. And so they were not able to separate necessarily just the collapsing glomeruli versus the normal looking glomeruli from the same patients. Nonetheless, they were able to see that there was a stratification in FSGS NOS and collapsing glomerulopathy when compared to normal controls or even minimal change disease patients. And so this was at least the first study to show that there were some distinct pathways that were occurring in collapsing FSGS, which they said was consistent with what's known as the dysregulated podocyte phenotype, which I'll describe in the next slide. Now, the dysregulated podocyte phenotype, this is a study by Vivek Dagadi and Laura Barasoni, where they actually used immunohistochemistry to try to understand what is going on with these very reactive looking epithelial cells that are capping the collapsed segment of the glomerulus. And that they found in HIV and collapsing FSGS, there's frequently an upregulation of mitotic markers like Ki67, and a loss of many other markers that we typically associate with podocytes like WT1, photocollexin, synaptopodin. So these cells are somewhat different. I don't know if we can even call them podocytes. They might be some kind of reactive epithelial cell that has some intermediate phenotypic characteristics. And this, of course, has also been seen in the context of regeneration from parietal epithelial cells that Dr. Shankland and Charlie have done a lot of work on as well. So in either case, at least using immunohistochemistry, it was seen that collapsing glomeruli have this abnormal phenotype. So that prior study that I showed to you with laser capture really didn't study viral-induced collapsing glomerulopathy. And so the question comes again, are the same mechanisms pertain to HIV and COVID-associated collapsing glomerulopathy? And also, are the mechanisms the same in these two viral-induced diseases? And so for this, we used a relatively new technology that now we have access to, which is called spatial profiling or spatial transcriptomic profiling. And the instrument that we're using is from NanoStrength, just down the street, called the Geomics DSP. So the workflow is shown here. So here we can affix a kidney tissue section, and this can be FFPE tissue, which is a real bonus here. And we hybridize RNA probes to that tissue. We then counterstain it with antibodies so we can visualize structures. And then we can selectively extract the gene expression signature using next generation sequencing from any structure of interest. So we can hone in on the glomeruli, for example, and we can get the transcriptomic signature at the level of individual glomeruli, which is a huge difference and huge advantage compared to the prior laser capture microdissection methods I showed to you before. There's also a very rapid workflow. So really in 4 to 5 days, we can go from a biopsy to actual expression data. The experiment we initially performed was done with about 1,800 genes, but now we can profile the entire transcriptome as well. So just to compare and contrast these two technologies, I presented to you before laser capture and then the new material. Powerfully compared to other technologies, this works very well in FFPE tissue without any prior optimization, and is tolerant of the degradation and cross-linking that can occur to RNA in FFPE tissue. The biggest advantage is that you can have data generation from individual tissue sections without pooling, so that our sensitivity goes up quite dramatically. And it also doesn't destroy the tissue, so you can actually post-stain it to see what the exact structure was that you generated the data from. So this is what the experiment looks like. So here's literally a kidney biopsy that we utilized in the study, where we actually took 3 normal patients, 3 patients with HIV associated collapsing glomerulopathy, and 3 patients with COVID, including this individual I presented in this case. And we performed this experiment on that material. We stained with a variety of markers that allow us to define different structures. And here you can see there's a glomerulus that's outlined, which we were able to capture. We also looked at the tubulointerstitium, and we were able to actually also capture vessels as well. Now, using the gene expression analysis, we were able to nicely see separation in this PCA plot of glomeruli versus tubules and vessels, which confirms that these 3 different regions that we captured had different expression patterns as expected for those structures. And then you can see on the right, after the experiment was done, we stained it with our routine Jones stain, and we could actually visualize the structures there. So what kind of inferences could we get from this technology? Okay, so here I'm showing you one gene here, keratin 19, and here we looked at normal glomeruli versus the 3 HIV patients which had collapsing features, right? So I told you that before we were not able to separate the collapsing glomeruli from the normal glomeruli in terms of their gene expression signatures, but with this technology, we can do that. So these dots represent the individual glomerulus level gene expression signature for this particular gene. We can see that the glomeruli that have a collapsing phenotype here in these HIV patients have induction of upregulation of the keratin 19 gene compared to those glomeruli from those very same patients in the same biopsy that do not have that collapsing phenotype and have a more normal histology. Okay, so here you can actually look at an individual glomerulus. So here's this glomerulus, which has this level of increased expression of keratin 19. This is literally what it looks like. The data was generated from this glomerulus. This is a post-stain after the experiment. It's got prominence of those epithelial cells typical of collapsing glomerulopathy. And here, keratin was also used in the antibody stains that we used during the experiment. So we can see that there's induction of the keratin gene as well, but at the protein level, shown here by the green stain. By contrast, the normal looking glomeruli from those same biopsies, which look like this, do not have any induction of keratin protein. So we were able to directly correlate at the level of individual glomerular structures, gene expression and protein on the same, within the single biopsy tissue. So we were able to confirm and extend some of the previous findings from that other laser capture microdissection approach. I'm not going to go into detail here. We saw a lot of genes that were overlapping as well with the previous study. We also identified many genes that were not identified in the previous study. And interestingly, many of these were downregulated genes, which perhaps were just diluted in the prior study because they had to pool normal and collapsing glomeruli. So again, this highlights this increased sensitivity of this method. So finally, I wanted to say, kind of return to the question, is all collapsing glomerulopathy the same, right? And the answer is no. There are certainly some common core pathways that are activated during the collapsing process in terms of increase of keratin expression, downregulation of podocyte marker genes. But there are also differences depending on the etiology, the specific etiology of collapsing glomerulopathy. So we saw that the patients with HIV-associated nephropathy versus COVAN actually had differential regulation of many genes, and most particularly in the context of interferon. So we saw that the HIV-infected patients, many of whom had ongoing active infection, had marked upregulation of interferon-induced genes, whereas the COVID patients who had cleared their infection did not have induction of these genes, even though both groups of patients had collapsing glomerulopathy, okay? And so that's again shown in the lower set of panels. Here are 3 genes which are canonical interferon-inducible genes. I'll call your attention to these kind of blue dots, which are again the glomeruli from the patients with HIV disease. You can see compared to the COVID patients and the normal controls that these genes are upregulated in the glomeruli of these HIV patients. But if you're paying attention, you can see that there are some glomeruli in these HIV patients that don't have induction of the interferon transcripts, okay? And so these happen to be from a single patient who was actually on retroviral therapy and had an undetectable viral load. So again, saying that this patient probably had collapse in the context of other probable risk factors because they didn't have an interferon-induced signature. Again, this is showing that there's a lot of heterogeneity in the way the glomeruli can present histologically and the gene expression signatures that they're manifesting. So really in that patient, without an interferon signature, but with collapse, can we really blame this on HIV? Or is this due to some other causes, perhaps APOL1 high-risk genotype? Should we be calling it HIVAN or should we be calling it something else? We noticed that even normal glomeruli in HIV-infected patients can still have an interferon signature, right? So this is showing the complexity of layering on molecular diagnoses on actual biopsy material, which I think could be a very exciting avenue for in the future. So really what I'm telling you is that sometimes the histology may not predict the molecular signature, and vice versa. The molecular signature may not always predict the histology. And perhaps in the future, we should be thinking about this where we combine this and label a particular structure and say what's going on in terms of the gene expression pattern to get a more complete picture of what's going on pathologically and also etiologically, all right? So I think we've definitely learned a lot from these cases. And I think it's been, this technology is coming online. We've got the instruments now at South Lake Union. We're hoping to get them fully activated by early next year. And we'll be very excited to collaborate with any interesting projects that you have in mind. All right, so I'll stop there. We have a couple of minutes perhaps for questions or comments. Yes, Brendan, you have a question?
Yeah, sorry for a naive question. FFP is paraffin fixed?
Yes, so that's our usual way of processing the biopsy. So formalin fixed and paraffin embedded. Yep. Yeah. So that's really the exciting part of this technology, right? We can go back to archival specimens. We can go to rare cases, right? So we can think about, lupus is a great example, right? I mean, we have this, again, another classification, which is morphologically based, but we know that not all patients have the same kind of inflammatory response or injury response within the podocytes. All the pathologists have seen cases of lupus, for example, encrusted with deposits, but without any, crescents or necrosis. So what's going on in those glomeruli? We can consider, Kelly's in the call here too, fibrillary GN and some other rare diseases. And we don't have to only focus on the glomeruli. We can also look at tubular interstitium, right?
How long do you have paraffin blocks archived, and how much material is needed for something like this?
Yeah, so by regulations, it has to be at least, I think, 10 years. Though in practice, we just keep the blocks forever in the warehouse. The beauty of this particular method is that you need only a single standard 5-micron section to execute the experiment. So it's really minimal amount of tissue that is required.
I was just going to add about the technology and what you presented shows a great detection and measure of an interferon signature, and that really, we use tubular reticular structures as a clue for that. We have to get an electron microscope to actually see that. Whereas this is really showing us some in-depth molecular signatures that point to that. And you brought up lupus nephritis. There's been a lot of effort to try to look at interferon therapies or targeting interferon to treat that disease. And I think that, this type of technology is maybe an inroad to trying to understand what is the active pathophysiology. And in patients with lupus nephritis, they might not all have, prominent interferon signatures. And so that might be an explanation for the challenge of getting these therapies to actually make some headway. So this, I think, is a great example of understanding molecular heterogeneity and where morphology falls short in terms of our ability to classify diseases and maybe guide or help guide treatment.
Yeah, I didn't show it in this presentation, but we also had a patient with, COVID TMA, thrombotic microangiopathy, that had a completely different gene expression pattern as well. So I think it's just, again, clearly there's also the caveat of saying, when we talk about biopsy-based studies, sampling is always an issue, right? So we don't know where in that disease process that particular patient may be. But nonetheless, it's showing us that when we think about it at the time of biopsy or the time of treatment of that patient, different mechanisms may be at play. And I think that's still important to think about in terms of what that means for therapy and prognosis for these patients. So Kelly and I and Charlie are super excited clearly about this technology. It's kind of where oncology has been for a while, but hopefully we can make inroads for nephrology here.
Thanks so much, Ram, and also David for presentations today. Amazing presentation, Ram, and thanks to everyone for joining this morning on an unusual Friday after Veterans Day. So we'll see everyone next week for grand rounds before we take a break following that for Thanksgiving. So thanks everyone and have a great weekend.